that we assume it's a hospital. So what are we doing in terms of training and our community, at least for the basic life support? We know we have a lot to do in the in-service training, but at least the community should be aware and able to support, strategically support these events. And the third one, which I think it's a major one, and we need to discuss this as a health system, we need to somehow to restructure our coordination of referral system, and maybe you have already started, tabling this talk with the responsible stakeholders, but we really need to see where the services are and where exactly we are referring this patient and for what.
Thank you. The last question, yes, you're on point. That's happening, looking at the referral. The first question related to pediatric emergency, yes, very, very important. We saw that we are supporting an initiative that's ongoing to start a pediatric emergency super specialization here, and I know that's at the very high level of the niche, but our hope is that will trickle down. Beyond that, what's going on at the moment is we have a dedicated pediatric emergency care training, and I'm making sure it's not pediatric critical care, emergency care training that's specific for providers in lower level, because not everybody can be a specialist, and care has to be provided as you're creating specialists. So definitely that's part of the curriculum that we have as a short course, and it's ongoing. The issue of community is very complex in our country for many, many reasons. One is trust. We've done a lot of work on that. I think the area that we are able to and we are going to work on right now is working with community health workers to start with, as well as the first aid responders that are under Tanzania Red Cross. Those are well integrated in our system. We go step at a time, and we move. We probably will reach the level of the family. Most of us, when somebody gets sick, you know what they do. I will stop there because of time.
I think we have to move on. One issue is that I would like to point out that if there are not sufficient resources in a country, you need to map where red spots are. Swala la equity with inadequate resources will not take us too far. So I think my advice to you is that map occurrences so that you can justify resource allocation. Thank you. Makofi. Now it's time to invite the next speaker, Professor Takamasa Ueno, the Director of Joint Research from Human Retroviral Infection, Kumamoto University, Japan. Karibu. I'd like to give a brief description of Ueno. He's the Director of Joint Research Center. As I said, he's also a Professor of Infection and Immunity and at Graduate School of Medical Sciences, Japan. But he also belongs to our department, Microbiology, Immunology, MUHAS. So he's my neighbor. We are neighbors. We share offices. Karibu sana. He's going to talk about SARS virus. So listen carefully, and then we have time to ask questions.
Anyway, thank you for kind introduction, Professor Mate. And then also, I'm very honored to give me such a wonderful opportunity to, like Professor Bruno, DVC, and Professor Nair, DLP, and organizing committee of this such a good conference. And before I'm going to talk about my scientific part, I want to introduce how I am, or I have been, getting very close to MUHAS. Just like one is I mentored 6 PhD graduates of Tanzanians and then also MUHAS. And currently, I am mentoring 3 PhD students right now, and also play a role as a co-survivor, like 2 MUHAS PhD students as a co-supervisor. And I will be, or another person will be welcoming to my lab to Ilumba Mkenda will be joining us in this October. And in addition, we have made a short course internship program, and I invited a number of MUHAS students to my lab so far. And also because of based on these activities, other labs also invite Tanzanian PhD students. Like at the moment, there are 4 Tanzanian students in different labs, and also 2 more MUHAS students will be joining in this October. So our partnership is massively expanding, and also like we are like contribute to capacity building activity to MUHAS. And finally, this is possible because of our strategic planning between institutions. We begin MOU at faculty level back to May 2013, and also expanding to university level 2016, and then continuing this MOU right now. And also we established liaison office in Tanzania at MUHAS. So our partnership is also like long-standing, sustainable, and keeping expanding. Okay, now I want to go to share recent our data about cellular immunoresponses to SARS-CoV-2 variants. And as Professor Mate introduced, I am also a visiting professor at MUHAS. This is a part of some MUHAS activity. So our achievements so far in COVID research is by focusing on cellular immunoresponses to SARS-CoV-2, we quickly adapt to establish SARS-CoV-2 viral infections, and then also analyzing human immunoresponses. We published so far greater than 10 publications, and we got like greater than 1,500 citations so far. And I'm gonna introduce some of the work recently we made. And looking at loads of cytotoxic T cells in SARS-CoV-2 infection, there are 2 different aspects of functions and activities so far reported. One is protective effects. It's not only neutralizing antibody, but many groups show like cytotoxic T cells have like protective effects in response to vaccine, and then also secondary infection of the SARS-CoV-2. And also, our major issue is associating with reduced severity. If we have a memory T cells, then it is really strongly correlates with reduced COVID-19 severity. So this is why we believe that CD T cell responses against SARS-CoV-2 infection is important, long-standing way to protect as well as like keeping health even in the presence of SARS-CoV-2. And how cytotoxic T cells recognize antigens and respond is there is a HLA class 1 molecule bound in viral peptides here. And this peptide binding group is highly polymorphic within one of the most polymorphic regions in human genome. And therefore, dependence on the genotype, the different sequence of viral peptides are presented by this molecule. And this is presents in the surface of virus-infected cells, and then CD8 T cells recognize the complex of peptides and HLA molecule, and so that they produce antiviral cytokines or dominant effects of directly killing virus-infected cells. And so we established assays to detect responses specific for SARS-CoV-2 immediately after the pandemic happened. So we collected blood from convalescents and also vaccine recipients, and prepare PBMC from these donors and stimulate with viral antigens, including overlap peptides or virus-infected cells. And then looking at antigen-specific T cells by T cell activation markers and secreting antiviral cytokines and also directly killing activity. And then we focus on one specific HLA-A24. This is because this molecule is one of the most frequent class 1 molecule all over the world, especially in East Asia. And we Japanese have greater than 6% of population have this HLA genotype. So, and then we focus on CTL responses specific for SARS-CoV-2 spike protein because we focus on vaccine recipients as well, and in the context of this HLA-A24 genotype.
Recording stopped.
Oh, unfortunately. Looking at CTL epitopes in the spike protein, we, because of IT technology, we estimates or speculate these three sequences can bind to HLA-24 molecule. And then experimentally looking at how these peptides can be an epitope for human populations having HLA-A24. And the results coming up this vaccine recipients and also convalescents. Unfortunately, it's this one epitope did not respond like this. It's just only a subtle effects on T cell responses. However, we are calling NF9 and QI9, is these two epitopes dominantly recognized by a number of subjects we collected. So therefore, we think these two epitopes are immunodominant epitopes restricted by HLA-24. And also, looking at other functions of such cytotoxic T cells, and one is also in a secretion of TNF, interferon, And then also this one is CD107. That's a marker of T cell degranulation, is associating of killing activity of the T cells. So we reviewed that the CTLs specific for spike epitopes are extremely polyfunctional. And then back to the epitope issue, so as I said, there are two immunodominant epitopes, is NF9 and QI9, but this is two distinct characteristics, because NF9 is located at receptor binding domain. This is the place where the bunch of mutations reported in response to neutralizing antibodies. But in contrast, QI9 is located in a highly conserved region of the spike protein. So we raise a question: do such mutations at receptor binding domain might impose secondary effects on CTL responses? For example, confer escape from CTL responses, not only from neutralizing antibodies. So looking at the NF9 regions, these are one is prototype, is we say Wuhan strain, and also this is a few months after pandemic of the SARS-CoV-2. There's reported it's mink farm in Denmark. It's reported of one mutation here, and also this is a Delta strain, is in leucine to arginine mutation here, and also this is Lambda strain, is emerged in South America in leucine to glutamine mutations. So looking at these mutations, what happens? And all of these mutations, as a result, is reduce T cell responses of all the individuals we tested. So therefore, all of these three mutants or variants, CTL lose or fail to recognize these virus-infected cells. So this is a kind of T cell escape mutations. And then it's as you know, it's newly emerging Omicron variants coming up soon after Delta strain. And we looked at the sequence of these regions and found there is no mutations within the epitope region in Omicron variants. However, one mutation outside the epitope in Omicron BA1. So the question we raised is such CTL can retain antiviral activity towards newly arising variants. To review these, to answer this question, we established a novel assay system to directly evaluate antiviral activity of cytotoxic T cells. So we isolate from patients of SARS-CoV-2 virus itself, and also this is a human lung cancer cell line, and introduce HLA-A24 and co-culture with cytotoxic T cells from vaccine donors, and then looking at viral replication. So the data shows like viral replication goes up in the absence of T cells, but in the presence of cytotoxic T cells, this viral replication is much reduced, something like this. And this is NF9 CTLs, and this is QI9 CTLs. So this is, we say, inhibitory effects by CTLs. And then the question is, what about the cases of, variants of concerns, and then also Omicron, and particularly Omicron variants? So looking at the first part of the graphs, this in the vertical axis is same relative viral copy number. So decreased viral copy number is meaning of higher antiviral activity. So looking at the prototype, and also like these are different, it's missing some of the data, but it's Delta and Omicron BA1. Or actually, this must be Omicron BA1, BA2, and Delta strain. And looking at these so in Delta strain, it's because of T cell escape, so no antiviral activity of CTLs are shown. But interestingly, BA1 is much more like higher antiviral activity for CTLs, even if there is no mutation within the epitope, only one mutation outside the epitope. And BA2 and also prototype shown here are looking very, comparably like inhibited by cytotoxic T cells. So this is the summary of the data from multiple donors. So one is BA1 CTLs enhanced antiviral activity. So two BA2 CTLs showed only comparable antiviral activity that's consistent with prototype infected cells. And to Delta is CTL lost its antiviral activity. It's as predicted by T cell escape by inducing to arginine mutation. So we really get interested in why BA1 is more susceptible to CTL killing. So one hint is actually coming from antigen processing pathway, because is like within the infected cells, an array of aminopeptidases plays an important role in like trimming of the epitope to a size, good size of as an epitope. So Omicron BA1 has one mutation outside the epitope. We hypothesized because of this mutation, so antigen processing pathway might be enhancing of presentation of this epitope. So to test this, we use like 3 different inhibitors that inhibit like different aminopeptidases. And then what we found is only this inhibitor statistically significantly affect the antigen processing pathway of BA1, antigen processing efficiency of the BA1 strain. So looking at this one, this is tripeptidyl peptidase 2 inhibitor, this one. So therefore, we confirmed the amino acid, aminopeptidase, this aminopeptidase play a role in antigen processing of this epitope in SARS-CoV-2 spike protein. And because of the mutation outside the epitope, that result in enhancing susceptibility for cytotoxic T cells. So looking at the two different epitopes, as I said, is there are, because of located in receptor binding domain, there are lots of mutations known in this region, and also including recently coming up the Omicron subvariants. But in QI9, it's highly conserved of this region. So any differences between these is our results showing NF9 is response is enhanced in BA1, comparable in BA2, but as no reaction made BA5, but some others are not known yet. But QI9 responses are all maintained and conserved. So therefore, as a whole, so cytotoxic T cell responses can maintain reactivity or that's antiviral functions even against newly emerging variants, including Omicron subvariants. So the summary and also conclusion of my talk is we rapidly established a novel assay system to directly qualify antiviral CTL activity, and antigen-specific or virus-specific CTLs show potent inhibitory activity to SARS-CoV-2 variants of concerns. And some CTLs show reduced or enhanced antiviral activity against SARS-CoV-2 variants due to mutations, not only within but outside of the epitope. However, that despite such observation, as a whole, CTL can also target conserved lesions and maintain entirely antiviral activity. So I believe these results highlight important roles of cytotoxic T cells in protection and control of SARS-CoV-2 variants of concern. And this is the acknowledgement and also my lab here. And it's special thanks to networks and also JSPS Quad program with MUHAS. And also, I want to it's just advertisement, is we have a plan to have a joint session in this afternoon, and 3 people coming from Japan to present their recent work. And also these are working on closely related or in collaboration with us. Please come to this session. And also finally, we are operating a scholarship program, and we want to have outstanding, not average, outstanding candidates welcome. Extremely outstanding candidates are welcome. So please contact Dr. Doreen Kamali, and she's gonna lay first screening and then shortlisted the candidates. And I stop here, and I'm very happy to take questions.
Thank you, Professor, for a very nice talk. I think it was easy to understand. Very easy. Now, I really appreciate the collaboration between Kumamoto and Tanzania and the platform that you're building here. I would like to invite Professor Bruno to make a comment on that, because I think this is really important.
Thank you very much, Chairperson. I'm really happy, and as always, Professor Ueno, MUHAS has been a home since 2016 when we signed the MOU when I visited the institution with Professor Liamuya. And ever since, 6 PhD graduates from MUHAS have completed their studies there. In addition, close to 10 faculties at MUHAS have visited the lab and got at least 3 months of hands-on training. And these are basically based on the funding that Professor Ueno has worked very hard to get. In addition to that, MUHAS has also received a number of equipment which are installed at the Department of Microbiology, the genetic analyzers, and many others that I may not remember the exact names, plus the storage capacities, refrigerators for biosamples. And through the support that the lab has given us, MUHAS was able to lead the nationwide HIV drug resistance survey, and a number of benefits that we reap from this. So, I mean, this is an open collaboration. Anyone who is willing to go through the easy stuff that Professor presented, that we all understood, you're most welcome. It's usually the call I usually post on the MUHAS media for anyone to ask and join the team. It's an amazing team. Japan is an amazing place to visit. I know Dr. Everyone who studied in Japan will agree with me. It's also a good place to extend your collaborations. There are various colleagues of Professor Ueno in various areas that he has graciously connected us with. So not only on this easy stuff, you can also do other difficult issues in various areas. So you're welcome again, and we are pleased to have this collaboration. Back to you, Chairperson.
By the way, Kamori is not a Japanese name, it's Tanzanian. I would like to invite one of the, yes.
Thank you, Professor Ueno, for a very interesting, easy to understand talk. I have two questions. You looked at the CTL immune response against SARS-CoV-2. So my question is, did you look at naive patients, the immune response, vis-a-vis those who have received the vaccine, and see if there's any difference on the immune responses between the two? And then number two, if there's any data which shows the decline in the immune response between the ones who are naive for the SARS-CoV-2 vis-a-vis the ones who have received the vaccine, if there's any difference in the duration of the decline of the immune response.
So, thank you for questions. And your question is naive or vaccine, or naive versus vaccine, or naive versus infected? It's
so. The first question is individuals who have been infected with SARS-CoV-2, the CTL immune response, compared to the CTL immune response in patients who have been vaccinated. So if there's any difference in the immune response. And the second one is on the decline of the immune response, again, between the two groups.
Okay, so actually it's one because it's, I mean, like naive is without infection or vaccine. It's one interesting question would be like pre-existing of closely active cytotoxic T cells in our body. And because it's in Tanzania or Eastern Africa regions, is I think SARS-CoV-2 infection is, did not cause a, like serious problem compared to some other regions. So we are actually looking at closely active T cells. Like it could be like MERS coronavirus or some other coronaviruses. And actually, it's now such a study going on, and the one recent interesting results coming up because of vaccine plus breakthrough infected in Omicron BA.5 is enhance antiviral T cells and neutralizing antibody responses compared to vaccinated only or infection only. So it might be something like combination of vaccine plus infection induce or activate our antiviral immunoresponses. And then the second question is, actually we know neutralizing antibody decline rapidly after vaccination or infection. Compared to that is T cell responses can sustain over 6 months or sometimes a year long. So therefore, induced T cell memory is very important to maintain our human resilience toward like SARS-CoV-2 infection, even though newly variants might be coming up later on. I hope this is these are answers to your question.
Yes.
Thank you, Professor, for the nice presentation, which is very technical but very useful for us. So my question is about spread of SARS-CoV-2 infection. In Tanzania, we saw that there's evidence of spread even in the first 6 months in rural areas where people have very limited contact with town centers where we could have expected they've met with other people with infections. But most of those patients were coming to the facilities with malaria, but with no symptoms of COVID. So we wanted to know, is there any link between exposure to malaria, maybe in endemic communities, and some form of protection to SARS-2 infection?
Is it on? Oh, really? It's a nice point. It's nice to see you again, Prof. And in that aspect is I saw some publications that infection of malaria or malaria infected people has relatively like or associating with reduced severity or no infection or no symptoms in COVID. So it would be very interesting to collaborate in the future with you. I don't have any answer right now.
I would like to invite Dr. Dorin to make some comments. I know you graduated from Japan and you are in charge of the molecular aspect in our department. Now, what are the challenges that you endure that you would like to be solved so that the lab can make an impact? I think this will be very interesting.
Thank you very much, Professor Mate, for giving me this opportunity. I'm kind of caught off guard, but the main challenges that I can comment that we face, at least in our settings, with regard to the molecular aspect of research, is that we are faced with challenges on infrastructure in terms of equipment. I think that is the major issue. And also another challenge is on human resource. We still have a limited number of expertise in the area of molecular research. Thank you.
I think in the interest of time, we'll end up here. Is it very boring? Are you going to get hurt?
Thank you, Prof, for a nice presentation. We have seen the CTL is a inhibitory in nature, as you presented. I would like to know, is it related to, is there any relationship with the clinical presentation of patients in terms of severity of presentations and the outcome of patients? Thank you.
Yeah, I can add on that. These studies have been done in Japan, where co-infections are not here. There's a lot of background noise in terms of multiple infections, and some of them will tilt the immune system towards the Th2 response. Would that affect the efficacy of the vaccine?
Actually, there are several reports like these are like association or relationship of like greater numbers of virus-specific cytotoxic T cells versus reduced severity or protective effects. Like for example, like health workers that remain uninfected in England has greater numbers of antigen-specific T cells in like a first year of pandemic. So, but right now is not only just the Th2, Th1 balance, but in the people in developed countries, it's mostly like in Japan, it's 70% of people are vaccinated. And also nearly like 40 or 50% of people are exposed to SARS-CoV-2 as in terms of like antibody response to nuclear capsid protein. So therefore, right now is very difficult to like distinguish vaccine or exposure or infection, which is more effective on these clinical symptoms. It's gonna be something like very much complicated situation right now.
So in the interest of time, I think we end up here. Let's clap for the Prof. I think this was a very easy topic to understand. Thank you very much. I think this our session comes to an end. I'd like to thank you all. Organizer.